Hello everyone, and welcome to a brief lecture on understanding mitosis. Mitosis is cell division, the division of somatic cells, mainly when we're talking about a human. Division of your skin cells, liver cells, bone cells, things like this. Mitosis can also be a form of reproduction when you're thinking about single-celled life. The paramecium, amoebas of the protist group, bacteria, all use this process basically for reproduction. Let's get started with its general purpose. If you're looking in something like the human body or any multicellular organism, growth is one of the major purposes of mitosis. We all start off as a single cell, which divides into two cells, then 4, 8, 16, so on and so forth. That's how we get to be a multicellular organism, be it a oak tree or a whale or a human being. That developmental process is now being heavily studied with the advent of stem cell research in recent years, understanding exactly how these cells begin to differentiate once they go through mitosis. Mitosis will always be there. It's what replenishes your skin cells today. It's how you get new blood cells all the time. So it's a continual process. Your stomach lining, basically a brand new stomach lining every 3 days. So constant rates of mitosis in some parts of your body, very little mitosis or none in other parts of your body. For example, heart cells do not go through mitosis. Once the heart is developed, those are the cells you have, and that's as far as it goes. Repair, if you think about maybe a cut and it reheals, of course, you cut your skin and then it magically just grows back. Some organisms take this to a higher level. Some salamanders, you can cut their entire arm off and they will regrow that entire appendage, which is pretty impressive and also being heavily studied. And the reproduction is just for some species a single-celled life, which actually dominates this planet. Quick look at the few pictures here, some dividing amoeba there for reproduction. Once again, just reiterating reproduction, growth and development, and then repair with the tissue renewal. Let's take a closer look at a chromosome. These are the little packets of DNA that are actually going to be separated during mitosis. You take DNA from this string-like state of chromatin and you wrap it around these little proteins called histones in this very complex coiling process. If you look at a single chromosome, it appears to look sort of like an X. You have sister chromatids, one stick over here and one stick over here. They are held together by a centromere. The centromere has in it a kinetochore, which is a little motor protein that's actually going to walk the chromosome down the mitotic spindle. This is the apparatus that you're going to grow out of microtubules, which you'll be seeing here in just a second, that the chromosomes walk down. Your goal here is mitosis is to divide this chromosome and get this one packet of DNA on one side of the cell and this other little sister chromatid on the other side of the cell. And then you split the cell down the middle, and that's the basics of mitosis. Looking at the phases, we start here at interphase. You see this is G2, the last step of interphase on your far left there. Of course, you go through G1, which is normal daily life, then the S phase where you copied your DNA, and now G2 where you're preparing for division. In a G2 interphase, you're starting to get the mitotic spindle up here with the centrosomes. Your chromatin, the DNA, has been duplicated. You still have a full nucleus, and of course you'd have all the other parts in the cell as well. Not shown here, but they're there. You start off with the mitosis with prophase and prometaphase. We can kind of lump those two together. Now, there's 4 major events here that you're talking about in prophase and prometaphase. One, your nucleus is going to disappear. You see the nucleus here, you see it gone over here, so the nucleus is going to degrade. The mitotic spindle is going to fully form. You can see these spindle fibers. This is basically the highways that the chromosomes are going to walk down. The mitotic spindles are actually going to attach to the kinetochores of your chromosomes as major events of mitosis. So you're losing your nucleus, your chromosomes have formed completely. The mitotic spindle has formed, and the mitotic spindle has attached to the kinetochores. All 4 of those events right here in prophase and prometaphase collectively. Move this back over. Metaphase can be summed up by just saying that the chromosomes line up along an imaginary plate called the metaphase plate. As you can see in the picture there, the chromosomes seem lined up right down the middle, and you can imagine them pulling in half, where it used to be thought that basically it's like a little rope come out here and pulled this one side over here and pulled that other side over there. So now we know it's the chromosomes that actually walk themselves down these little mitotic spindle fibers. So these you can picture like a little highway that the chromosome is going to use to walk itself down. Of course, made of protein, as is the kinetochore. Go figure. Anaphase is the separation phase. This is where there's two major events. The centromeres of the chromosome divide and the sister chromatids separate. So in anaphase, you've got a centromere division, so both sister chromatids will now have a centromere, and the sister chromatids are going to walk themselves down to the opposite ends, effectively cutting in half, your DNA here, so that at the end of mitosis, you had 46 human chromosomes. You double that to 92 during the S phase, so you're going to cut this in half and you get 92 individual sister chromatids over here and 92 sister chromatids over here. When you pair those up, that's 46 and 46, so that's brand new human cells, equal amounts of chromosomes, both having 46. And then the last step, remember, of the cell cycle was cytokinesis, where you have the division of the cytoplasm. And in the telophase, what's going to happen, our last phase, is the chromosomes are going to begin to unwind and go back to chromatin, and the nucleus is going to be reforming around here. It's another view of mitosis, as you can see, using a fluorescent microscopy. And you look at prophase here, and you can see the distinct nucleus, and you can kind of see the chromosomes forming. The mitotic spindle's been highlighted here so that you can see it. If you look over here in prometaphase, see the nucleus is gone. The chromosomes have fully formed. The mitotic spindle is fully formed. Got your centromeres or centrosomes on both sides, centrioles. You look at these images, 10 micrometers across. Here's the distance. So very, very, very tiny, of course. Metaphase, they're kind of all grouped up there in the middle. The chromosomes are, and you can see the distinct, microtubules here of the spindle apparatus. Anaphase, you see two distinct bunches of chromosomes, one bunch over here, one bunch over here. This is where the centromeres have divided and the sister chromatids have separated. And in telophase, you can't really see chromosomes. They're unwinding back into chromatin, and the cell's got a cleavage furrow. It's about to pinch in half. And that would be the end of mitosis and the beginning of cytokinesis, the division of cytoplasm, which ends your cell cycle. It's a quick kind of close-up view of what's going on. If you look, this would be the kinetochore inside the centromere chromosome. You can see these spindle fibers actually attaching to it. You can see this is, pictures half a micrometer. So once again, extremely, extremely tiny. Give or take about 1,000 micrometers across the thickness of one hair. So this would be half of one of those. Another blown up view showing the kinetochores and the kinetochore microtubules. That's the attachment and the movement of the chromosome down the microtubules. Kind of a rendition here of what's actually going on. The microtubules made of proteins called tubulin, which are easily broken down and also easily reform. And the kinetochore you consider is basically like a little motor protein here that's walking itself, opening up and closing and pinching itself all the way down the microtubule, so that the chromosome itself, sister chromatid in this case, is pulled towards one end of the cell. So in anaphase, this is what's going on. You got a centromere pulling the sister chromatid this way, and you'll have the same thing happening over here with sister chromatids going in the opposite direction, and you eventually get the DNA cut in half. And then the last phase, of course, cytokinesis. Once again, different in plants and animals. Oak trees, definitely a lot of mitosis around here. They're very big in our area, but they have to build a new cell wall because they cannot pinch themselves in half, whereas our cells are more like bubbles and just sort of pop in half.